do me a favor and break down what does DNA stand for?
Deoxyribonucleic acid.
Did not see one coming.
I knew that one. It's awesome. It's
And RNA is.
Ribonucleic
I
acid.
didn't say that.
So close. Yeah. The best way to think about PCR is it's a DNA photocopier.
Okay.
And that's really how we can work with so little DNA, right?
Right.
We take these tiny, like 5 or 6 cells or tiny little blood stain or these, and we take it and we put it in our DNA photocopier,
Yeah.
and we take that small amount and we copy it 26, 30, 40 times depending on which protocol you're using, which gets you billions and billions of copies of DNA. That way we can take that and move it along in the process. And we can work with that level of DNA, but all it is just taking that small amount, making photocopies of it, making photocopies of photocopies, and it just expands out to massive numbers, which now we can actually visualize and we can get those profiles.
Do you inspect everything under a microscope, or do you have a machine that looks at it for you? Or?
DNA, I mean, there are the cool tricks that you can actually visualize DNA, but for the most part, it's too small to even see. So we've got instruments who detect it for us.
Gotcha.
So in a lot of this, as Derek mentioned, we quantify. We get an idea of how much DNA there is there. We find 3 things at that point. You know, how much DNA, how much male DNA is in there, and the quality of it. Is it degraded? Is it old? Is it how easy is it to get a profile out of it? At that point, we can make decisions on what to do. And there's a lot of different ways we can go, but most of them end up in that room right over there. Instruments that detect DNA. And what we're doing is putting little fluorescent tags on the DNA. And they are just colors, red, yellow, blue, I mean. And then we have an instrument that essentially all it is a tube, a very, very small tube, with a little window on one side. And we put in something about the consistency of corn syrup. And then we shove the DNA through, and bigger stuff goes slower, and smaller stuff goes faster. And it separates it out, and then we just detect what color comes up. And then that gives us a profile.
So, Josh Powell, if he was trying to get rid of Susan's clothing or some of her belongings, what would a person do to eliminate DNA? Bleach?
Bleach is a bad one. I would say it's very difficult to truly remove all the DNA.
You have to soak it all.
It gets into cracks and crevices. It's just difficult to remove completely. That being said, bleach is a bad one. Bleach is the one thing, and you see it on CSI. You do it. Right. It will mess up our process. And
so soak bones in bleach, where you just take all the DNA out of it, or would
Nope.
I just soak them while?
So actually, one of the first steps in our bone processing is actually bleaching the outside surface, because I don't know if you guys have touched the bones.
Never.
I know, right? I would never. I mean, you guys definitely had gloves and everything.
Absolutely, yeah.
100%.
Yeah.
I call them
And
comments.
and I would imagine probably dripping some sweat out there. And
They went through a process. not the DNA process.
So there's probably some, we'll call it contaminating DNA on the outside surface. We're going to bleach that outside surface, and we're going to get rid of all the contaminating DNA. And then
You hear that, viewers? Everybody who's been mad at us for manhandling the
They've
bones.
been so mad.
They've been like, you're ruining the crime scene.
I'm gonna forensic.
These guys are gonna fix our mistakes, so calm down.
So we're going to get rid of the outside surface, and then we're going to grind it up, and the DNA is all in the inner surface.
So with a bone, can you cut it, run the DNA, and then know quickly if it's animal or human? Like, is human and animal DNA way different?
So at this point, we'll know. So the DNA quantification that Derek just described, that's only human. So if there's DNA there, then we're going to detect it, and if it's animal, we just won't see anything.
Do animals have DNA?
They do. We
But
just
it's not
don't have,
that your system doesn't pick up.
animals, plants, everything. Everything is outside of viruses. But our system is human specific.
DNAs are inside the cells.
Yeah. So you've got
These guys are probably
Two
like,
copies.
what did you guys learn in high school? Nothing?
Not much.
You guys know-
And we don't watch CSI, so yeah, we're a bunch of rookies.
Well, one of the things that we do, one of the jobs that we have is to testify in court.
Right.
And so we have got to educate juries
Right.
about what the DNA process is. And
Yeah.
you can't expect to remember all the stuff from high school. Come on now.
We at least pass the class.
There'll be a test.
Kinda.
They don't teach that when you
Some
do your
of
GED.
You get jet.
But I mean, yeah, you have your billions of cells in your body.
Yeah.
Each one has a copy of DNA. It's a blueprint to build you, and it's coming from one copy from mom, one copy from dad. So you actually have two copies in every cell. And that is our process is specifically looking for human DNA. So if there's bacterial DNA, which there's going to be on some of these bones, or if the bones are animal, we won't detect that at all.
Right.
So we'll only see if there's human there, and that at that quantification step that says, all right, that's where we can stop with a lot of stuff.
Yeah.
If it's actually animal, or if it's just so bleached up that there's just nothing left.
Yeah.
But we'll know at that point. Here's how much DNA is there. It's human. It's this much male,
Yeah.
and it's this degraded, and we'll be able to go through the rest of the
So
process.
one thing I'm